olfactory sensory neuroscience that happened in the early 90s by Richard Axel and Linda Buck, who went on to win the Nobel Prize for this. And so this is just a diagram, first of all, of these are vertebrate olfactory receptors here. Oh, that's okay. And then these ones on the right are from flies. And basically, I just wanted to show this to illustrate the fact that vertebrate olfactory receptors are G protein coupled receptors. They are related to rhodopsin, as well as some other chemoreceptors found in worms. They are actually not related by sequence to the receptors that detect odors in Drosophila. Okay, so this is a bit of a weird thing. Flies have a completely, independently evolved set of receptors for odors, yet the organization of their olfactory system is very similar to the mammalian olfactory system. And so we use the fly model to understand principles of olfaction, even though the receptors are actually fundamentally different in their sequence. And they are probably not GPCRs, although there's quite a lot of debate about how they actually work. Interestingly, for any receptor aficionados in the crowd, one of the things that makes fly receptors really strange is that their topology is inverted. So normally the N terminus would be outside and the C terminus is inside, but that's opposite for Drosophila olfactory receptors. So I don't want you to kind of dwell on the details of olfactory signal transduction. I just want to illustrate briefly that is kind of a classic G protein coupled receptor signal transduction mechanism with a little bit of an interesting twist. So here is the receptor protein here, which binds to the odorant molecule. And then there's this heterotromeric G protein, including a G alpha that's specific to olfaction called G-olf. And so when G-olf is activated by the conversion to GTP, this binds to and activates adenylyl cyclase, which produces cyclic AMP, which opens up a cyclic nucleotide gated cation channel. And so then the flow of cations into the cell will depolarize the cell, right? That's all very typical and normal. The one sort of interesting peculiarity about this is that the calcium, when it enters, actually then activates a calcium-gated chloride channel. But ECL for these neurons is around 0. Okay, so they tend to have a higher intracellular chloride concentration than a normal neuron, and so it's about equivalent to the extracellular concentration of chloride. And what that means is that because VM is less than ECL, this drives an inward current, it depolarizes it, and because ECL is above threshold, it's actually excitatory, and so this helps maintain the depolarization of the neurons. Okay, so it illustrates here chloride flowing out, but of course it's an anion, so that indicates an inward flow of current. Yeah?
Sorry, is this, do these neurons work by action potential firing, or is it greater potential?
Yeah, they have fire action potential. It's a good question. Any other questions? So one of the really interesting things about the olfactory system and about olfactory receptors specifically is their genetics. And I don't want to get too deep into this because, it's not a genetics course, but this happens to be the largest family of genes in the mammalian genome. Okay, so mice have about 1,000 of these OR genes, these odorant receptors. We have about 300 because the other 700 have been mutated to become pseudogenes, which essentially means that they're still there, but they can't code a functional protein. So these are huge, huge families of receptors, and they're clustered within the genome. So this picture is just the mouse genome and showing each of the chromosomes, and then the bars indicate the sort of density of OR genes in those particular regions. And so you can see that there are particular regions that have a lot of these receptors in them. And this is just an example of one of those regions with all of the different OR receptors that have been duplicated within that region to produce so many of these receptors. And presumably, the reason why we have so many of these receptors is we need to discriminate between so many different odors, right? So lots of different ligands that you want to detect and tell the difference between means that one of the mechanisms you can achieve, or you can use to achieve this, is to have a lot of different receptors, and that's part of the answer for mammalian olfaction. And then things get really interesting when you look at the expression of these receptors. So these are just cross-sections of the olfactory epithelium. Which is abbreviated OE. And what these are showing are in situ hybridizations, which just show the expression of a particular gene, and they're co-labeling for two different ORs, MOR28 and MOR10 in the case on the left, 28 and 83, and then 10 and 83. So the pairwise combinations between these three different odorant receptors. And what they observed when they did this was that any cell that expressed one of these receptors never expressed the other one. Okay, so there was complete non-overlap between the expression patterns of any pairs of these odorant receptors that they looked at. Okay, and this has been extended now, because now we can do fancy things like single cell sequencing or sequencing of, I guess, the mRNA from a single cell, which has verified that individual odorant receptor neurons, ORNs, OSNs, olfactory sensory neurons, individual neurons express only one olfactory receptor. Okay? So that sort of spawned a really kind of interesting line of investigation into how that actually occurs. You've got 1,000 different genes that all have to be expressed in one tissue. They are expressed intermingled so that they're not like in little clusters or anything. You can see that here, right? Like each one of these is expressed all over the place. Yet they're never expressed in the same cell. So how does that actually work? And the problem isn't solved yet. I'm going to show you an experiment that's old at this point. It's about 17 years old. I guess it was done in 2000. But it illustrates a really sort of cool result, I think. And this has particular sort of relevance for me because this is an early sort of scientific memory of mine. So the guy who, one of the two people that discovered the ORs, Richard Axel, right, who's like this really famous guy in olfaction. And he came to give a talk when I was a first-year graduate student, and we got to like talk to him and whatever, right? But in his talk, he showed this result, and it blew my mind at the time. You know, my naive sort of first-year graduate student mind was completely blown by this result. And in retrospect, you know, maybe it shouldn't have been entirely, but I still think it's sort of a pretty cool idea. And so the experiment was this. They made a transgenic mouse that had the promoter for one of these OR genes hooked up to LAC-Z. Okay, so basically, this is analogous to kind of the fly stuff we talked about before, right? So we just have a LAC-Z gene, and they just put the MOR28 promoter in front of it. So they made one of those mice, and then they made another mouse that was the same thing, so it was also MOR28, but now this one was hooked up to GFP. Okay, so they could stain for LAC-Z in red, and they could stain for GFP in green, and just look at the expression pattern of these two things. So they looked in 2 different mice, each with one of these things, and of course they just saw, you know, some population of olfactory sensory neurons that expressed LAC-Z or GFP, depending on the genotype of the mouse. But these are using the same promoter, right? So then the cool thing happened when they made a mouse that actually combined both of these constructs. So this is the same promoter. So everything that we sort of know about gene expression, or in like a simplified way, should tell us that these should be expressed in the same cells, right? They're using the same promoter, they're in the same animal, so why wouldn't they be expressed in the same cells? But it turns out that they're not. Okay, so when they looked at this, even though these were both driven by the same promoter, they were never expressed in the same neurons. Some of them would express the LAC-Z version, some of them would express the GFP version, but no cells ever expressed both. And so this illustrated that it wasn't that, you know, each OR gene just had some specific promoter that drove expression in some specific set of cells. There must be some sort of overarching mechanism that selects just a single gene to be expressed in any one of these different neurons. Okay, and it's beyond the scope of this course to talk about exactly how that works. There's been a lot of work on it since those days. There's chromatin stuff involved and a bunch of stuff. It's actually much more complicated than even it appears on the surface. All right, so any questions about that? Yeah.
Are all of the pseudogenes that we have in our genome, are they still being expressed as if they were normal genes? So that,
Yeah.
like, some of the neurons are actually not smelling anything?
Yeah, that's a really interesting question. So, to directly answer your question, no. So every one of your olfactory sensory neurons, I believe, should be expressing a functional receptor, okay? To sort of extend your question to a little bit of a more nuanced variant, could you get expression of a pseudogene, like a non-functional receptor, along with a functional one? And that is an interesting question. I don't think so, because I believe that the expression of any functional receptor inhibits the expression of any other receptor, including pseudogenized ones. The other thing that I'll say is that many of the pseudogenes probably don't even produce a receptor protein that can get to the membrane. They're probably truncated and stuff. So I think the answer is basically no, that you just have one receptor expressed and it would be a functional one. Any other questions? Okay, so this brings us to the really important principles underlying the organization of the olfactory system. Okay, so there's basically 2 things that you have to remember about how the olfactory system is organized. One is the thing that I just said. Every olfactory sensory neuron expresses only one OR. Okay? Interestingly, it's not just one OR, it's actually one allele of each of one OR. So like you have 2 alleles of every gene, right? One from your mom and one from your dad. And every neuron will actually only express either the one from your mom or the one from your dad. But anyway, that's neither here nor there, really. So that's rule number 1, which we just talked about. This image shows rule number 2, which is that if you have a single class of olfactory sensory neurons that expresses one particular receptor, let's say this is all the MOR28 neurons, which I don't think it really is, but that doesn't matter. We can just imagine it is, that they all project their axons to one point. Okay? This is called a glomerulus. And so all of the receptor neurons, or all the sensory neurons expressing a particular receptor, converge on a single glomerulus in the olfactory bulb. It's actually two. For our purposes, I don't care whether you think of it as two or one. Conceptually, it's easier to think of it as being one, but the truth is the olfactory bulb is like sort of duplicated. There's a lateral region and a medial zone, and like it actually projects to a glomerulus in each of them. But conceptually, they all target the same little glomerulus, right? And so each of the corollary to that, the sort of flip side to every sensory neuron that expresses a single receptor converging on a single glomerulus, is that every single glomerulus is targeted only by the neurons expressing a single receptor. Okay? And so this is all illustrated here. So all the purple neurons project to this glomerulus, whereas all the red ones project to this glomerulus. And not only that, but any single glomerulus only gets targeting from one color of the sensory neuron, right? Where the colors here represent an individual receptor gene being expressed. Yeah?
So we have as many glomerulus as ORs?
Yes, exactly right. So you have as many glomeruli as you have ORs. So that would be like 1,000 in the max. 2,000 if you want to count the duplicated versions. So the important thing here, which we'll get into in more detail as we go on, is that this allows the olfactory bulb to represent odors with each glomerulus being essentially a functional unit for representation, right? So it's taken this very disorganized periphery in the epithelium where, you know, any given receptor is expressed in all these neurons that are sort of scattered throughout, and it, through convergence to these individual glomeruli, it's causing it's making the map more discrete, right? Whereas where activation of one receptor will cause activation of a single glomerulus. Okay, and this is really important for understanding the coding of odors in the olfactory bulb. So any questions about that? Yeah.
I'm still a bit confused about the two glomeruli. Is it
Yeah.
two, one doesn't matter? Oh.
I mean, it's two. I struggle with this. The reality is it's two, but for our purposes, conceptually, it's easier to think of it as being one. And it's not so unrealistic to think of it as one because, like, you've got one group where you've got one set of glomeruli where every one is a different receptor neuron, and then you've got another set. There's like a duplication, right? So within every region of the olfactory bulb, there's one glomerulus that's targeted by an individual receptor. That's basically it. Any other questions? Okay. So this image just compares the organization of the sensory neurons in mice, which is what we just said, right? Every neuron expresses only one receptor. To flies, which conceptually, I'm going to say basically a similar thing again, conceptually, this is also true. So you can see, ignore this neuron, I don't know why they show it, it's like a complete outlier, but this neuron would express the purple receptor, this neuron would express the light blue receptor. One peculiarity of the fly olfactory system is that all of them actually express a second receptor, okay? And this is an obligate co-receptor. We don't have to worry about the details, but so every individual neuron will express the dark blue receptor and then plus one specific receptor, okay? So functionally speaking, the odors that a neuron responds to is determined by that specific receptor, so the purple one or the light blue one or whatever. The dark blue one just has to be there for functional purposes. In worms, That is not the case. So worms have about 800 receptors, and they only have 300 neurons in their total nervous system, right? So obviously they can't have one receptor per neuron. So in C. elegans, sensory neurons express many receptors. So this chart is just summarizing kind of the diversity of receptor genes in these various animals. So there's lots in mice and humans and C. elegans. There's actually not so many in flies. There's only 60. And this has been a really important reason why flies have become a really powerful model for looking at olfaction, because it's like an order of magnitude simpler than the vertebrate olfactory system. Okay, and then this is just what I just said, individual receptors in each neuron and the type of receptor. All right, so I want to show briefly the organization of the fly olfactory system. This is a fly brain on the right, and on the left is a fly head, and on the fly head is labeled in different colors, different receptors being expressed in the two tissues or the two organs that it used for smell. There's the antenna, which is by far the more well-studied and important of the olfactory organs, and then there's this second organ called the maxillary palps. Okay, most of what we talk about will relate to the antenna. The neurons from these areas project to a structure called the antennal lobe, which is right here in the brain. This is exactly analogous to the olfactory bulb in mammals. Okay, so you can see the axons from a single receptor neuron population targeting the single glomerulus in the antennal lobe, and then as well they project to the other side to target the same glomerulus on the other antennal lobe. In flies, everything is completely hardwired, so people who study olfaction can just look at the 60 glomeruli and be like, okay, I know which one each of these is and like what odors they respond to. Okay? So everything is completely pre-specified. There are about 50 olfactory sensory neurons or olfactory receptor neurons, ORNs, same thing, that project to an individual glomerulus, right? But the organization is the same. So all the red ones are projecting to the red glomerulus, all the green ones are projecting to the green glomerulus.